we'll be looking at how the actin cytoskeleton is organized, regulated to make some of this stuff happen. So what's the key mechanisms behind the involvement of actin in all of these processes? So what are those processes? First, movement. Okay, phagocyte needs to be able to move towards areas of tissue damage or areas in which pathogens have potentially got into tissues. So they need to be able to respond to the chemical signals being released at these locations in order to find the cause of the damage and carry out phagocytosis of those pathogens. So movement or migration, cell migration. Okay, now cell migration requires extension of the cell at the front, so to move forward, but retraction of the back of the cell in order to generate translocation, so to move from one place to another. Okay, so in order to get cells to be able to migrate, you need cell shape changes that allow that to happen. You need the membrane to move forward at the front, and you need the membrane to be retracted from the back. Migration. Phagocytosis. Once the phagocyte reaches the pathogens, it needs to be able to extend its membrane around them so that it can engulf them and carry out the process of phagocytosis to form the phagolysosome in order to get the breakdown of those pathogens and preventing spread of those pathogens throughout the cell, or to be able to display those antigens on the surface to initiate the immune response. So to be able to do that requires extension of the membrane around the pathogens. So phagocytosis. We also have cells, so many cells in the body need to have a high surface area in order to carry out their function. Things like hepatocytes, or liver cells, which carry out detoxification, need to very efficiently be able to absorb material or harmful chemicals as they move past them in order to metabolize them, convert them, or detoxify them, and then send them back out into the fluid. So hepatocytes need to have a high surface area. In the kidneys, you have the cells of the proximal convoluted tubule, which absorb useful things and prevent useful things from the filtrate, preventing useful molecules from being lost from the body in the urine. So the proximal convoluted tubule also needs a high surface area. Those structures increase their surface area by having microvilli. Now microvilli, in order to get these structures, you need to be able to extend the membrane, beyond the normal boundary of the cell. Okay, in other things that the actin cytoskeleton also does. So we've talked about membrane extensions, but contractility is also another role of the actin cytoskeleton. So this contractility is important in migration, pulling up the back of the cell. Contractility is also important in cytokinesis during cell division. So in order to get the cytoplasm or the cells to pinch in the middle, in order to get that membrane to come in the middle so that you can create those two new cells after the cell division process, you need to contract the cell membrane at that point. That involves the actin cytoskeleton. And finally, the most famous, if you will, of the contractile structures, the sarcomere of muscle cells. Okay, so smooth muscle, cardiac muscle, skeletal muscle, they all work on the same basic principle of using actin and myosin fibers or filaments to slide against each other, generating that contraction of cells that results in contraction of muscles that results in pulling forces being generated. Okay, so there we have like the summary of F-actin, filamentous actin involving processes. Now let's look at the structure of actin filaments and see how some of this stuff is done. Actin really is made up of, or actin filaments are made up of the rather, structurally kind of uninteresting globular protein. So it's the protein itself is called actin, but the subunit of actin is called G-actin because it's globular. Okay, so we've got the G-actin subunit, if you will. Now the subunit, it polymerizes to form, so you've got many of these subunits that join end to end to form this filament. Okay, but in fact, in the final filament that is produced when the subunits polymerize, it kind of looks like an intertwining of two of these. So let's have that kind of come over the top of that, like so. And then going behind like that. So that's essentially what filamentous actin look like. So in this, when actin proteins are like arranged like this, then we call it F-actin or filamentous actin. Filamentous, that's the globular protein. Okay, so the globular proteins join end to form the filament, and the filaments combine as a pair to form the final full actin filament right there. Now what we are going to do is represent this in a particular kind of way, because filaments are polarized. So filaments are polarized, so they have one end which is more actively growing. Okay, so we'll call this the plus end, and there is the other end is less actively growing. Okay, and we're going to call this so that's the plus end right there, and the minus end is at the other side. Okay, now because these proteins are joined end to end, and because they have this, all of them have the same orientation, so they're all kind of pointed in the same direction. When you look at the structure of the fiber, it has an overall, it has polarity in two ways. Is this really important? Yeah, so the overall fiber, the overall filament has a polarity in two senses. In one sense, it's got the property that it's faster growing at one end, but it's also got the property that it's got an overall shape polarity as well. So overall, it's got a barbed end. So monomers join like this. So it's got a pointy end, it's said to have a pointy end, and a barbed end. Okay, so that same filament right there, look, can be represented like this, with this end being the plus end, so it's growing faster at this end, but also in terms of structure and appearance, it's also said to have the barbed end on this side. So this and the other side, not only is it the slower growing side, but it's also called the pointed end. Now remember, I'm only including that detail because it's going to have some relevance later. Okay, so that's the overall structure of the filament. Now, individually, these filaments are not very strong, but it is their organization and their collective structure that is what has impact on the cell shape. Remember, it's the actin cytoskeleton essentially that has got the biggest impact on the regulation of cell shape. So any cell shape changes that are involved are done through the F-actin cytoskeleton. So as we have discussed, the actin cytoskeleton needs to be able to do two things. it has to be able to cause membrane or cell extension, and it needs to be able to generate some kind of contractility. All right. Now, first, how does the filamentous actin network, how does it cause any change in a membrane shape? So the first thing is this, that as we've said, the binding proteins of these cytoskeletal elements are very important. So the F-actin fibers polymerize near the membrane. Okay, that's fine. Okay, but the key way in which they influence or are able to influence cell shape or what the membrane is doing is because they have actin binding proteins that attach them to the membrane. So you have these proteins that attach the ends of the actin filaments to the membrane. Okay, so what that means is so these proteins that connect the F-actin cytoskeleton to the membrane then link the shape of the cell to whatever's happening to the actin cytoskeleton. So if the actin cytoskeleton is being polymerized and it is pushing out on the cell membrane, these attachment proteins allow the polymerization of the actin or the polymerization of actin, the extension of the actin network, to cause the extension of the cell membrane. But if the F-actin cytoskeleton is being organized in a way that generates retraction, these attachment proteins allow the membrane to be pulled in along with that process. So these attachment proteins are a key part to understand how the actin cytoskeleton influences the membrane and therefore the cell shape, both in terms of extension and retraction. Okay, so now let's look specifically at the extension and contractility processes. In terms of extension, so let's say we have a cell membrane right there and we need to generate some extension from it. Now what happens, or what is thought to happen, is that in terms of extension, what needs to happen is that you have the barbed ends of the actin fibers facing the cell membrane. So because these barbed ends, we discussed that these actin filaments have a polarity, and the faster growing end is the barbed end. Okay, so I'm just re-representing that here. So when this is the situation, when the barbed end is organized toward the outside and there's active polymerization on this end here, then that means that as the polymerization happens, the cell membrane is pushed from within by the extension of, or the polymerization of the actin networks, which means that where there's more active polymerization, the cell membrane is being pushed outwards. We have contractility. Now, contractility is really interesting, but it is comforting to know that many of the contractility processes involving actin happen in pretty much the same way, whether it's the sarcomere, cytokinesis, or contraction at the tail end of a moving cell. Contractility involves the binding protein, or the actin binding protein, myosin 2. Now, myosin 2 is a protein that's got a kind of coiled region and a head region there. The head region, so let's just redraw that. Okay, so that's a tail region and a head region, and the head region is what binds actin filaments. Its head region, so if we have, first thing is that these myosin molecules, they will bind actin fibers, but they will bind in a particular orientation. Okay, and minus, plus, plus. Okay, so if we have an actin fiber like this, its minus end is here, its plus end is there. The myosin 2 molecules bind in a particular orientation. They bind, and the head is able to bind the actin fiber, change shape, and push the actin fiber along in the direction, in a particular direction. So they will bind, change shape, move it, detach, bind again further down, push it along, detach, bind further down, push it along, detach, and so on. That cycle continues until, you know, we got a significant movement of the actin fiber relative to the myosin, or the myosin moves relative to the actin fiber, whichever one is fixed. Now what happens during contractility is that you have these myosin molecules, but they're arranged in opposite directions. So you might have one in this direction, and you'll have another one with its coil this way, but its head facing the other way. And on this side here, you'll have an actin fiber, but in the opposite orientation. So while you have this one plus to minus, on this side you'll have the polarity of the fiber in this direction, i.e. its plus end is here, its minus end is here. Now this myosin 2 molecule is binding on this actin fiber and moving it in that direction. Okay, so what's happening is we've got two actin fibers, and they are being pulled or moved in opposite directions. Now, membrane attached at either ends of these fibers would be being pulled in to generate contraction, be it during cytokinesis or being contracting behind the nucleus, allowing the cell to move forward. Okay, so I mean, to summarize, I know it's probably too late to say that to keep things simple, but whenever you have actin fibers in the same direction next to each other being polymerized, then it causes the extension of the membrane wherever that's happening. So active polymerization of all these actin filaments, polymerization in the same direction collectively generates enough force to push on the membrane and cause its extension. But whenever you've got the anti-parallel arrangement of actin fibers and the presence of myosin 2 to cause the sliding of those filaments against each other, that is what is required to generate contraction through the actin cytoskeleton. Okay, now what we'll do is finish off by just looking at the different kind of structures you can generate using these mechanisms. So extension structure, so a migrating cell would need, so let's look at that migrating cell, the nucleus right there. Okay, we need extension at the front of this cell. Now extensions happen when you've got these branched actin networks at the front of the cell. So you've got extensive branched networks, but importantly, these fibers are all kind of the plus ends are at the front right here. So these branched actin networks are forming these structures that push on the cell membrane, causing the cell membrane to extend forward. Their plus ends are all at the front right there, so they're all generating force in the same direction. Okay, so these kind of cell extensions are called lamellipodia. So they result in cell extensions like this. They might also be responsible for the extensions that result in phagocytosis. Okay, so if we've got the nucleus there, we've got some bacteria right here, it will be branched actin networks that will be pushing on the cell membrane in order to get these extensions. Okay, and the really magical, almost magical thing about this is that we discussed that it's cell signaling and all that regulation. So the cell signaling happening that's influencing the formation of this branched network. So the bacteria will be releasing chemicals, potentially these chemicals will be binding to cell surface receptors on these cells. The receptors will then initiate signaling events inside the cells that will activate actin binding proteins that are responsible to reorganize the actin cytoskeleton into these branched networks. Thereby, these membrane extensions are formed only when those signals are encountered, which is cool. Okay, similarly, in order to get this cell in a position to be able to do this, again, those signals are causing the extension of the cell at the front via similar mechanisms. But in order to get contraction at the back, remember, we've got actin filaments in opposing directions with their plus ends in opposite directions, and the presence of myosin too, causing them to move in or slide in opposite directions to get that contractility at the back and help this cell move forward. Okay, so we've got lamellipodia. Remember, maybe these are called uropods. Okay, so we got that, and we also discussed that you have cells which have microvilli to increase their surface area. Well, microvilli are nothing more than, instead of a branched network resulting in cell spreading and broad extensions, we have finger-like extensions caused by actin bundles. Again, they all are similarly oriented so that they push on the cell membrane in the same direction. Okay, but when you have these bundles of actin filaments, rather than broad extensions, you have these finger-like projections which can be used to increase the surface area of the cell, allowing it to enhance its function in some way, depending on the function of the cell. Mainly, this is because of transport functions. Okay, was there anything else? And again, the contraction, the actin myosin, you have actin fibers in the sarcomere. You've got these actin fibers. Okay, then what causes the contraction of the sarcomere against the myosin is that you have the myosin heads on the thick filaments in the sarcomere, binding to the actin fiber, pulling it along, binding and pulling it. So the same mechanisms apply in causing the contraction of the sarcomere. Am I forgetting anything? So we've got lamellipodia, uropods, microvilli. And we're forgetting cytokinesis. So in cytokinesis, I'm kind of running out of space here, but in cytokinesis, what we have is rings. So we have rings of actin filaments, but whereas some of those filaments are going round in one direction, you have others going in the opposing direction, and you got myosin 2 in between, pulling them in opposing directions, causing constriction in the middle part of the cell because of that, and eventually you have cytokinesis happening, splits the cell into two thanks to the F-actin cytoskeleton. Okay, so I hope that has maybe shed a bit of light on the mystery of the involvement of the cytoskeleton in these processes. The next thing we'll discuss is the intermediate filaments, ladies and gents. So there are a number of things which come up in A-level biology in which the cytoskeleton is involved. Which processes slash structures actually, which processes slash structures involve the actin cytoskeleton? That will be question number 1. And the second thing would be, can you describe the structure of actin filaments? Describe the structure of actin filaments. 3, and this is going to get progressively a little bit harder, but can you explain how the actin cytoskeleton, how actin cytoskeleton generates both membrane extension and contractility, right? Because that's its kind of key things that it does: generate cell shape changes that result in extension and contraction. So can you explain how that is done? And finally, can you briefly describe the actin, or describe the actin or F-actin organization required to generate the different structures, processes. Okay, so if you can do these things, then you probably know at least the actin part of the cytoskeleton inside out, back to front, much better than you need to know it for A-level. But yeah, okay, so as you watch the video, think about these questions, and maybe it will help you do a review at the end. If any of this stuff is not clear from my video, then that should be the basis of the questions that I want to get from you guys. Remember, questions is the way that you know you are learning. No questions, no learning. That's what I say. Okay, so think about these things, and then if any of this is not clear, that means some part of what we have discussed has not set itself nicely in your brain. That means we need to talk about it further. Further input, further research is necessary.